It's a pleasure to be here, always is. I have learned to enjoy the warm associations and relationships and intellectual camaraderie that I've found here over the years. And I had known Bob Nerum actually before he first moved here. And when I found out he was here, that was yet an additional stimulus for me to come. And we like to joke that we actually had our first formal working and collaborations together. We were in a hotel room in Las Vegas working on a NSF grant together. So it's sort of a strange way to start, but it has been wonderful working with folks over here. And I sit on the board of the Institute, and when I come over and see what's going on, I'm always awestruck. And so I think this is one of the most wonderful relationships in American academia, and I thank you for having me. One of my major interest, well, I've been interested in blood vessels for 25 years or so, and actually got into interest in blood vessels through a meeting a fellow named Mike Gimbroni, who many of you may know or certainly know his work. I met Mike also in at least a gambling environment. We were playing poker when we were both at the NIH one night and started talking about blood vessels, and actually left and walked out and established what became a long relationship. Mike, incidentally, was one of the people who was first interested in describing the effects of physical forces on endothelial cells and how they signal, and actually established a collaboration with Forbes Dewey, who's in mechanical engineering at MIT, and published some of the early works in that field. My focus, I'm also a clinical cardiologist and try to be scientist. And we've been interested in the clinical disease of atherosclerosis. And what I'm going to do is summarize kind of actually quite a number of years' work contributed to by many people I'll mention as we go along. And at Emory, we have been focused on oxidation mechanisms as a internal signaling system for at least about 10 years. And that brought several things together. And this whole business of oxidation and the use by multiple cell types, in fact probably all cell types in the body, of signals that are generated by the stimulated production of reactive oxygen species is coming to be understood to be a signaling mechanism or second messenger system in very much the same way as cyclic nucleotides or intracellular calcium are. And that is reflected in the wide appreciation that multiple human diseases actually have important elements that are reflected in generation of reactive oxygen species, and frequently are reflected in the fact that at autopsy or at biopsy that one can see terminal oxidation products of either oxidized proteins, carbonyl groups, or lipids that in fact reflect that in the disease process that there's excessive generation of reactive oxygen species. And I just update this list periodically. The et cetera is kind of making room for the next iteration of it. But you can see that there are a large variety of the common diseases that afflict us have some common mechanistic features, and that's going to be one of our themes. This is kind of a primer on reactive oxygen species, and one of the principal or initial reactive oxygen species or radical that is formed by multiple enzymes and including the mitochondrial metabolic pathways that is not shown here is a generation of oxygen anion or superoxide by this one electron reduction. And that can be metabolized by superoxide dismutase, which is one of the key degradative enzymes to hydrogen peroxide, which has the potential through interaction with divalent cations and other mechanisms of forming actually much more reactive species than is the oxygen radical. And also not included on here, I will allude to it later, are nitrogen-containing radicals that include some nitric oxide itself, which we'll talk about, is a important radical. In fact, it's a powerful antioxidant, but nitrated products or nitrogen-containing products include some very toxic radicals that can affect biology. But one can, through these pathways, generate lipid radicals, hugely change the biology of the cell membrane simply by filling it up with lipids that in fact become very reactive species themselves and affect signaling in fairly profound ways. Now, atherosclerosis has, incidentally, I'm sure all of you know, is the most common cause of death in our society and indeed in most Western societies. And it has three major characteristic features, and how you divide this up is always in the eye of the beholder. Ones that I've already made some oblique allusion to that has been a focal area of interest here for a long time is the non-random distribution of the disease. That in fact areas in which there are low shear stress, disturbed flow, oscillatory shear, as we'll come back to, are sites of predilection for the disease. It's non-random and tends not to localize, at least initially, in areas where one has laminar non-disturbed flow. Another feature of it is that the disease is, or characteristically, is an inflammatory disease, and one of the earliest manifestations is the recruitment into these areas of predilection of mononuclear cells and also, and somewhat less appreciated, T-cell populations. And then one develops an inflammatory milieu, and that this localization of inflammatory cells and the changed metabolic milieu also results in very extensive remodeling, and the ultimate in remodeling of disruption of the structural integrity of the arterial wall, and resulting in acute thrombosis, myocardial infarction, stroke, or remodeling in the other way, which I'll show actually some of Zeno Gallus's slides, leading to aneurysmal formation, dilatation. But basically, the fundamental process is remodeling. This is an article, actually a beautiful slide of Peter Libby made in a review article in Scientific American a few months ago. And the characteristic features that you see here would be the normal artery up here, and a thickening, and then the recruitment of mononuclear cells, T cells, and one of the key manifestations of the abnormality here, in fact, is the expression at the endothelial cell surface of adhesion molecules that interact in a lock and key fashion with counterligands on the mononuclear cells and T cells. and then these move into the arterial wall, take up LDL, which becomes oxidized and is not taken up by the mononuclear cells until it is oxidatively modified. And that observation, the original observation of that fact, which was one of the important advances in understanding this disease, was made to an important extent by Sam Parthasarathi when he was in San Diego, but is now on the faculty at Emory. And these accumulation, called it foam cells, which produce a large number of cytokines and chemokines that enhance the inflammatory response, that one gets plaque progression, gets a fatty core here of these foam cells, one gets migration of smooth muscle cells into this thickening area. And then ultimately, to an important extent, through the secretion of various matrix metalloproteases, one gets structural disruption of the integrity, exposure of the flowing blood to this lipid core, which, among other things, contains large amounts of procoagulant tissue factor. That leads to thrombosis and clinical catastrophe frequently. That's shown also here. Now, and this whole notion of understanding this disease, although the historical antecedents go back to actually at least to the 1930s, when a fellow named Timothy Leary was working at the Boston City Hospital and made observations about macrophages in atherosclerosis, and it was his son who was the drug guru of the 70s, who died in 60s and 70s. I've forgotten what it was. Tune up, something tune out, taking whatever the drug of the day was. He was a temporary Harvard faculty member. But his father actually was one of the contributors to the notion of this basic disease as an inflammatory disease. That began to be put together intellectually in the mid-'80s, and it became apparent began to be really apparent in the early '90s that this process was fundamentally related to the clinical events of plaque thrombosis and rupture. And that point was made very clear in a paper that was published in Circulation in 1994. And this is from an autopsy specimen of a patient who had died of acute myocardial infarction. And on the right side is a computer enhancement of this upper panel. There's an elastin stain. This is a lipid core, and you can see this fracture site, and the elastin has been, and connective tissue has been degraded. That led to the exposure of blood to thrombus. This is the residual lumen here, thrombus. And then if you take the adjacent section, stain it for macrophages, inflammatory cells, you see this intense population of inflammatory cells. They're right at the fracture site. And the smooth muscle cell, there are no smooth muscle cells immediately in this environment, which are cells that would ordinarily give structural integrity to the plaque. And so that began to really stimulate that line of thinking then began really to stimulate a great deal of thought about what there is about the disease that recruits white cells into the arterial wall and how that begins to act. Now, immunologists and people interested in the basic aspects of inflammation had worked out, mostly in the microcirculation, a lot of understanding about how white cells are recruited to areas of injury and stress many years ago. This slide kind of summarizes, it's actually for lymphocyte but monocyte nuclear neutrophils and other cells are very much the same, and that one has G protein coupled receptors in these cells that interact with ligands on the arterial wall, adhesion molecules that then these become active, these receptors become active, recognize, this would be a chemokine, for example, and this is an adhesion molecule. You see the stimulated expression here, lock and key, and then arrest and diapedesis and movement of white cells into the arterial wall. Now, that notion, which was well established even in the 80s and aspects of it even back to the 70s, what was also known from work in a lot of laboratories that if one starts high cholesterol feeding in an animal, the rabbit was a typical example, that one could see within a few days the beginning of this localization and sticking of macrophages at these sites of predilection in the arterial wall. And the mechanisms involved were really not known, and how did the endothelial cell know that the cholesterol was high, for example. Mike Gimbroni, who I've already mentioned, who started actually working on cultured endothelial cells, one of the first people to culture endothelial cells, and worked with Judah Folkman, and who was interested in tumor angiogenesis, one of the fathers of the field of vascular biology, as is Mike. But they had been working with cultured endothelial cells, stimulating them with cytokines, seeing that macrophages that they put in would stick, and demonstrating the expression of new proteins on the surface that mediated that interaction, developed antibodies to these new proteins that were expressed to separate out antibodies, took the rabbit models that of high cholesterol feeding, looking a few days later, seeing mononuclear cells attached, and then began to ask questions of what was going on using their reagents that they had developed in vitro. And they had, and then what they saw with one of their antibodies after just, and this is an artery from a cholesterol-fed rabbit, just a few days after beginning a high cholesterol diet. And what one sees, this is a macrophage right here, just under the endothelial surface. One can see others up here, the macrophages here. This brown stain here shows the results of interacting and staining this wall with one of their antibodies, and so everywhere they saw a macrophage sitting immediately under an endothelial cell that's just kind of moved in, taken up residence, they saw this staining. You can see that right there. They identified that had specificity. They could see it with one antibody. They called that, since it appeared, and they could block these things with antibodies, and so they called it atherosclerosis leukocyte adhesion molecule, first called it atheroelam. It became apparent that that molecule was the rabbit equivalent of a well-known human adhesion molecule called VCAM1, vascular cell adhesion molecule 1, which was known to be involved in the attracting of white cells and T cells into the arterial wall. Again, that had been established earlier by immunologists. And so this paper, and this work was actually going on when I left the Brigham in 1988, but was eventually published in Science in 1991. And this I've already alluded to is VCAM1, which is not constitutively expressed except in areas, you can see it expressed in areas of abnormal hemodynamic forces at branch points that you can see this molecule expressed in linear areas of the artery. Normally it's not expressed at all. But it is expressed in the high cholesterol state, and then it interacts and through mechanisms that we've already talked about with these receptors on the arterial wall that have been activated by chemokines. So that was known in the early 1990s. This just is a slide from Cy Wilcox at Emory that shows in a human fatty streak and these are macrophages under the endothelium, a stain for endothelium, and showing the expression of VCAM1 in this human specimen. And then the question that Russ Medford and I asked, and Russ is maybe known to many of you, was an assistant associate professor with us, became involved in a company that we formed around these observations, a company called Atherogenesis, and Russ is now a very successful businessman, president and CEO of that company that actually came out of just the work that I'm showing you now. But the original question, which was asked over beer and cokes over a few months, and we were trying to put together an idea for a program project grant, was basically the question at the bottom of the slide. How does the endothelial cell know that it's in a high cholesterol environment? You could substitute diabetes, you could substitute smoking, you could substitute hypertension, as it turns out, because they're common features. But if you could answer this question, then maybe we could make some progress, which is what the challenge that we had before us. Now, you'll wonder how I got from there to here. And this is a coronary angiogram, but I'll tell you in just a moment. Some years earlier, when I was still at the Brigham, we became interested in endothelial function, the vasodilator function of endothelium. And that is one of the major mechanisms of keeping blood vessels relaxed and indeed keeping them quiescent with respect to coagulation or inflammation, was in fact dependent upon a phenomenon that initially was called endothelial dependent relaxation. And then that was described in a very important paper by Robert Furchtgott in about 1981 in Science or Nature. And the fundamental observation was that blood vessels would dilate in response to the muscarinic cholinergic agonist acetylcholine, as it turns out a lot of other things, only if the endothelium were present. And if you scraped off the endothelium, then that didn't happen. It was something that was transmitted from the endothelium to the underlying smooth muscle that turned out stimulated an enormous amount of interest. Turns out it was, at least to an important extent, nitric oxide. The discovery of nitric oxide came from that, which stimulates guanylate cyclase, among many other things, and cyclic GMP, and dilates blood vessels. But, and subsequently resulted in 3 Nobel Prizes about 3 years ago, one of which went to Robert Furchtgott. But we were interested as clinical scientists in that kind of phenomenon because of the clinical manifestations of coronary artery disease in terms of coronary spasm. Is that we had known, or people had known since the late 1970s, early 80s, that diseased coronary arteries, one cause of myocardial infarction was in fact sudden squeezing down, spasm, occlusion of the arteries. And pathologists had been telling us for 20 years, even by the 1980s, for 20 years, that whatever's wrong with arteries in this disease is a problem with the endothelium. They knew that because of multiple models of Russell Ross and other people who would scrape the endothelium off, feed the animal high cholesterol, and just see this, these lesions develop like crazy. And so the dictum in the field was that you had injury and the Russell Ross's response to injury therapy, and the assumption was that you were losing the endothelium, and then that's why people got disease. There was only one minor thing wrong with that hypothesis, is that when they looked at specimens, the endothelium was usually there, except in these fast stages. So that actually led to the development of the term of endothelial dysfunction, which Mike Gimbroni was actually important in, so something's wrong with it, but we don't know what it is. That was kind of 1980 or so. Then Robert Furchgott, who was a pharmacologist, came along with the observation that there's an important vasodilator function of blood vessels, the endothelium, that is endothelial dependent. So we said, well, whatever the pathologists are talking about, maybe it would be reflected in the clinical events and the vasospasm that I was mentioning to you earlier. So after many months of talking to the Human Investigation Committee at the Brigham, they finally let us do studies like this. Now, these aren't the original studies, but shows the same kind of thing. These are actually from Arshed Kuyami, who has now joined us at Emory. But it's a coronary arteriogram with a catheter you can see going up here from the groin, around the aortic arch, and into the left anterior descending coronary artery. Now this is not, and you see this is just a pacemaker here, because they're going to inject acetylcholine, which was the drug that first got originally used in the lab to show either dilatation or constriction. And this is not a normal coronary artery, and this is the left main coronary artery, and the left anterior descending here. And then you see, but you can see irregularities right up here. And then this, there are minor irregularities there. But actually it looks pretty, generally pretty good. And these are pretty normal arteries. And so if you just compare, the size of this artery and this artery, and you infuse acetylcholine down this infusion, you see that this, in fact, gets, if anything, a little bigger and dilates. But it certainly doesn't constrict. But on the other hand, if you take a patient with atherosclerosis, and you don't see a lot of atherosclerosis here, but they have atherosclerosis other places, maybe in the right coronary artery, you instill acetylcholine, and look what happens. Virtually disappears. So that was the kind of data, and we actually published those kind of data in the New England Journal in about 1985, and kind of implying that this might be substrate for the spasm we talked about. If you compare in the same vessel the response to acetylcholine, which in fact is stimulating the endothelium and stimulating vasoconstrictor with no development, with no decreased release of nitric oxide, the vasodilator. But if you substitute with a drug called nitroprusside, actually delivering nitric oxide, then you see this dilatation here. Now, I'm going to move forward a little bit faster to get you through the points of the story. But the bottom line with what was going on in thinking about this was that, in fact, the metabolic problem in endothelial cells that was associated with that abnormal vasoconstriction involved, at least in part, the excessive production in this disease state of oxygen-free radicals, which was known to interact with nitric oxide, degrade it, actually form a bad actor called peroxynitrate, but that the vasodilator defect actually could be explained, and these are from experimental animal studies of Dave Harrison at Emory, that you could explain a lot of this on the basis of excessive presence of reactive oxygen species, oxygen radicals, that are degrading the nitric oxide and not allowing that. So then the question gets back to, back to the question of the VCAM and that inflammatory molecule, and the answer to, or at least the hypothesis that we proposed and thinking about it, Russ Medford and I and others with whom we were discussing it, could the VCAM1 that is stimulated to be expressed on endothelial cells in atherosclerosis, could the signal be oxygen-free radicals? And so we actually posed that as a hypothesis to ourselves initially, because in the early 90s there was evidence in prokaryotes of genes being regulated by oxygen-free radicals. And so we actually did, and we knew that the VCAM gene had promoter sites, or sites in its promoter that bound NF-kappa B. There was beginning to be some evidence in T cells that this could be inhibited by a compound called pyrrolidine dithiocarbamate, which is a powerful antioxidant. And indeed, in the experimental culture systems, we tried this, stimulated endothelial cells with cytokines, and saw induction of VCAM1 and other cytokines, and showed, in fact, that we could inhibit it by a powerful intracellular antioxidant that interfered with this signaling mechanism. And this is a slide from an article describing this in the Journal of Clinical Investigation in 1993. And these are northern blots from human endothelial cells of VCAM1, E-selectin, and ICAM1, other adhesion molecules. And you can see stimulated with interleukin-1 beta, and you can see this is control, that you see a powerful induction of E-selectin, VCAM1, ICAM1, and that only the VCAM1 was inhibited by the antioxidant, giving evidence of some significant selectivity and redox sensitivity of this transcriptional control pathways. That led to a lot of other things, but so the question, one of the questions was, is this really related a function of VCAM1 itself, and we're showing that, in fact, and I'll show you in just a second, that you could take the same compound, put it in animal models, and see protection against atherosclerosis. And we'd assume that VCAM1, and still assume that VCAM1 is one representative of a class of redox-sensitive pro-inflammatory genes. But the fundamental question was, is there something that's fundamentally important about VCAM1, or is it just a surrogate for a whole class of things? Well, Cebulski, Myron Cebulski, who's no longer at Harvard but is in Canada, actually has continued to study VCAM1 over the years. If you knock the gene, the knockout, the VCAM1 knockout, is an embryonic lethal. So there's something required as has to do formation of fetal membranes. And so you couldn't study that in an atherosclerosis model. But what they did is did a partial rescue of that by a deletion, and the VCAM has several immunoglobulin-like domains in it, and they modified one of those so that they could get partial expression of VCAM1, and animals survived. And then they bred those animals into a genetic background that produced atherosclerosis. So, and with the LDL receptor knockout. And then, so in that case, they could look and see in the VCAM rescue with that expressed 8-10% of VCAM on the cell surface, would that affect atherosclerosis? The answer is yes. You see the VCAM wild type here, and you see the staining. This is Sudan 4 staining probably, and you see, and I don't know whether you can see very well, but at almost every intercostal up here that you can see the red staining. You can see it along here. In this partial rescue, you see a dramatic difference, and that was so there was a gene-dependent relationship, gene-dose-dependent relationship with susceptibility to atherosclerosis, and VCAM, ICAM1 knockouts didn't do anything much at all, just suggesting that in fact VCAM was an important player. This doesn't project very well, but some of the early experiments that were actually done at the company showing a rabbit aorta, and this pink is VCAM1 expression, that is inhibited by putting the same kind of antioxidant in the drinking water, and showing, and these animals have cholesterols of 1,500 or something. They actually don't live very long, and you can see the same kind of protection right here. And these are very high cholesterols, but you can decrease the inflammation simply by this same kind of pretty toxic molecule. We actually had some stupid idea at one time that might be a drug, but a drug that comes from a compound, a family of compounds that have uses, among other things, as fungicides and curing rubber, is not likely to be a good drug candidate, but we didn't realize that at the time. So, but at any rate, the molecular mechanisms showed possibilities. Now, we're going to go forward, and I'm going to go through expansion, the form of, expansion of the artery in general, but narrowing the formation of a big plaque and the ruptured plaque has been associated with varying expression of these family of matrix metalloproteases. And this is a slide that really I thought was remarkable and led to my initial interest in recruiting Zarina to Atlanta, which actually showed from a human specimen, carotid endarterectomy of taking a atherosclerotic plaque, removing it at surgery in some of the carotid disease, putting it on a slide, staining it for these metalloproteases, stromolysin, gelatinase. These correlate with MMP, something which I've gotten to the stage of believing in neuronal synaptic conservation, so I haven't remembered which these correlate with. But if she also put the substrate for these enzymes and exposed that, and then this is reflected, this brown staining here is reflected, in fact, in enzymatic activity in these human plaques, which really relates directly to this remodeling and potentially disruption. And what she showed subsequently in more recent observations, suggesting that the foam cells that I showed you earlier that come from these plaques are robust producers of reactive oxygen species. And if she looks at specifically MMP9, these are secreted in the extracellular space in a proform, and that the reactive oxygen species are actually involved in the activation mechanism and actually generating the activity that causes the remodeling of the artery. This is a slide of a human coronary plaque from Bob Taylor showing really intense production of reactive oxygen species right at the shoulder of this plaque that we know is a highly vulnerable area. This is my miss and not Zarina's because the computer didn't handle it right. But the bottom line is that the reactive oxygen species, oxygen radicals, that are produced by foam cells actually are intimately involved in the activation from proform of these matrix metalloproteases. And then getting to kind of the third fundamental characteristic, this is a very old slide, and I wouldn't be surprised if there was someone in the audience that said, that's my slide, but if it is, I thank you. It is 12 or 13 years old, or I've forgotten exactly where it came from. But shows the fundamental phenomenon I was talking about earlier, that in fact the areas of susceptibility of the plaque formation in fact are opposite to flow dividers, and these are the areas in which VCAM may be preferentially expressed early on. It's areas of low shear or disturbed flow. And then that is illustrated diagrammatically here with the branch points. See the vulnerable areas opposite to the flow dividers as you get a disturbance of flow. This is a model very much like the carotid artery, which is an area of great clinical importance in terms of plaque formation, and that one can get in this carotid bulb where the internal and external carotid branch, and you just, by the way it is, that you get disturbed patterns of flow, and that's one of the most common sites of atherosclerosis development that's of clinical importance. Formation of these lesions leads to clot going upstream, and that's a major cause of stroke. But these disturbed flow patterns are critically important to that. And we published a paper showing that oscillatory shear stress as contrasted with laminar shear some years ago when David Chappell, who actually did a lot of his work here, was with Bob Narum and worked jointly in the program, showed that and showed that it actually affected adhesion of molecules. This is an update of those kinds of studies by David Harrison, actually, and his group published a paper using ESR and using more up-to-date technology, showing that in fact, if you look these are shows with non-sheared endothelial cells, the superoxide signal. One get, if unidirectional shear, some modest increase in that signal. But in fact, if you put a reversing component into it, you really kick up the oxidative stress. And this just shows a summary. And that with oscillations, disturbing flow, you're actually turning on the cells to produce increased reactive oxygen species. And then that would then fit in the general hypothesis that I've been developing. And this is a slide from David Chappell's work of about 5 years ago now that show that if you stimulate cultured endothelial cells with the cytokine IL-1, you get, here would be a stagnant flow or no flow. You get a robust stimulation of VCAM and other adhesion molecules that if you grow the cells in with a laminar shear of 5, then in fact that inhibits that cytokine stimulation, suggesting that the laminar flow, normal flow, in fact is creating an antioxidant environment. The selectivity there gives some truth to that. And this just summarizes that the local hemodynamic environment modulates susceptibility of development of atherosclerosis. Laminar non-disturbed flow creates an antioxidant, anti-inflammatory environment. We actually know a fair amount about the molecular mechanisms of that. It's a powerful inducer of eNOS. Nitric oxide is a powerful inducer of superoxide dismutases and the counter-regulatory ions, enzymes. And disordered oscillatory flow is pro-oxidant and pro-inflammatory. Now I'm going to I'm going to summarize something else quickly, and then go get to the last point and close. But there has been known that there is a relationship between the development of clinical atherosclerosis and the renin-angiotensin system from the kidney and elsewhere that controls blood pressure and volume. That has been known from clinical observations for 30 years. And people, major clinical trials using drugs that block that system. ACE inhibitors, angiotensin converting enzyme inhibitors, one of the most widely used drug in the world, blocks the conversion of the angiotensin 1 to angiotensin 2. And these are useful drugs developed as antihypertensives, but also useful in people with heart failure and with major heart attacks, and improves remodeling of the heart. And people really did not suspect strongly that those would be anti-atherosclerotic drugs. But some of the big clinical trials that appeared in people that had heart attacks or had heart failure, that in fact they were having fewer of these events that of acute myocardial infarction and stroke. So they were suggesting that the renin-angiotensin system in some way also was pro-inflammatory in the context that I was describing earlier. And the mechanism of that pro-inflammatory response is actually shown in kind of this Christmas tree looking slide here, and from cultured cells. And this is work from Kathy Grinling and people in her group, showing that angiotensin 2, which is a business end of the renin-angiotensin system, actually is a robust stimulator of reactive oxygen species and hydrogen peroxide, which is shown here. We showed, or Kathy showed, that in fact angiotensin 2 stimulates an NADPH active oxidase, and this is a signal demonstrating that angiotensin 2 is coupled to a specific enzyme, the NADPH oxidases, that through the model we were talking about earlier would expect to be pro-oxidant, begins to provide some linkage to the general model that we were building. There's a lot known about the structure of these oxidases. There are now large families of them. A lot of the work has been done by Dave Lambeth, and I'm not going to go through this other than to say that the enzyme in endothelial cells that is involved in the phenomena that I was just showing you with the ESR spectrum is in the same general family as the phagocytic enzyme that is involved in bacterial killing in phagocytic cells. And this just shows that if you put this enzyme in a nude mouse, that it causes tumors, and just shows its potent biologic activity. And this just summarized that the anti-inflammatory effects of the drugs that I was describing to you are probably being effectively antioxidants because they're blocking the stimulation of these vascular oxidases. If there's a long history of interest in vitamins and antioxidant vitamins, and in fact the most common ones are vitamins E and vitamin C, have been pretty ineffective in clinical trials. And that seemed to be a paradox, and two possibilities: either the drugs are not right, or the whole notion's wrong. And one of the most dangerous things in science is to begin to get severe emotional attachments to your ideas. So you still have to always say. Actually, I frequently tell people that one of the first experiments you ought to do is the killer experiment, the one experiment that would say the whole notion is wrong. So sometimes you can save a lot of time. But still, the possibility was: is it wrong, or are those just not the right drugs, not potent, whatever? There was evidence, a drug called Probicol that is no longer on the market here or in the U.S. because of side effects, was actually developed in a large screening looking for antioxidant compounds by Dow Chemical in the early 60s. This molecule, which has these phenol groups up here, and this linkage. If you take half of these, and it's beta-hydroxytoluene, which if you look on any label, look on your bread wrapper, and you'll see it contains BHT as their antioxidants. But you put these together, and this actually lowered lipids, but it also has adverse effects, and was taken off the market here. And I will come back to that in just a moment, because part of what atherogenics became was actually using this chemistry and then turning loose some very talented organic chemists on it, and they came up with something new. I'm not going to talk about the statin drugs, but just to get to the point. We had developed the company, which became public a couple of years ago and is moving forward. Actually made that Probicol molecule asymmetrical with succinamides, and changed its biologic properties very, very dramatically, its bioavailability, its solubility, where it goes, and made a good chemical a lousy drug into what potentially is a good chemical and good drug, which is the whole idea. And there were a lot of preliminary data. The FDA made the company do a year-long study in monkeys, a hypercholesterolemic monkey model, just to demonstrate whether or not some of the adverse effects that had been seen in limiting, the parent molecule Probicol would actually be a problem in a primate model. It had gotten through the usual toxic screens. So this had monkeys on a high cholesterol diet for a year, actually fed Probicol, which has some antioxidant activity, and what you see is just this is a coronary artery, and you see the development of a pretty robust plaque. On the other hand, this is AGI 1067, which is a modification of that I was telling you about. And this artery in this primate model shows effectively no atherosclerosis after a year. It was highly protective. And then this business, that's kind of where we are today. Actually, the story is a bit further along now, in that this drug that showed that in monkeys has just been given go-ahead to move into phase 3 of clinical testing in humans after having a successful phase 2 trial in Canada. And so then this just kind of summarizes the notion of oxidation and atherosclerosis and antioxidant therapy revisited. And the mechanisms by which antioxidants inhibit atherosclerosis may not be solely due to their ability to inhibit the oxidation of LDL, which was the central dogma for about 15 years. Therapeutically important features may be due to direct alterations in metabolism, function, gene expression of vascular and inflammatory cells, and then redox-sensitive transcriptional regulatory factors may be attractive therapeutic targets. So that then kind of goes from notion, in vitro testing, in tissue culture, animals, people, and what at least the data thus far suggests that in fact you can view a lot of aspects of this disease in the paradigm of enhanced oxidative stress in the arterial wall, and that at least is out there as a potential unifying hypothesis, and that therapeutic agents targeting that mechanism then are moving forward. Thank you very much.
Are there any questions?
Yes.
Any thoughts about the antioxidant targets on cells, whether they might be intracellular or extracellular?
Well, presumably a lot of them are intracellular, which was kind of the strategy that we had taken, because if you look at Probicol itself, in just the slide I showed you, at a good dose is a very good antioxidant. It circulates and is bound to LDL. It affects the oxidation. If you do oxidation of LDL in vitro, it affects the delays, increases the lag time in oxidation of that molecule. What it doesn't do is get effectively inside cells. So, and that and a lot of other evidence suggests that it's intracellular compartments that become oxidized and change protein conformation by affecting disulfide linkages, among other things, and either have a disulfide linkage or non-linked sulfhydryl groups. And so you change conformation and thus change protein activity, and there's a lot of evidence for that. And these families of enzymes that have been studied, there are a lot of other oxidases. We focused on the NADPH oxidases, but those are distributed very specifically at different sites in the cell. A lot of them actually interestingly in the perinuclear region, but they are very specifically targeted. That's a large. Just that oxidase system is a large family. That at least 7 members of that family have different distributions, different cells, but basically are intracellular enzymes. And so a lot of the action is intracellular. Some, the activation of metalloproteases is extracellular. Perhaps a lot of the degradation of the nitric oxide may also be extracellular. Yes?
There's been a number of clinical trials of MMP inhibitors for arthritis and tumor cancer. I'm just wondering if any of those clinical trials they have looked at maybe the effect on arterial disease simultaneously.
I don't know the answer to that. I know that there's still a lot of interest in metalloprotease inhibitors. I know there's been some toxicity associated with some of those drugs. Actually, as it turns out, just this morning I was looking in Nature Cell Biology reviews and on their website and saw a summary of an important new pro-inflammatory mechanism that in fact is very dependent upon the cleavage of syndecan, and I've forgotten some of it out of my area of expertise. But the whole notion was that at least one MMP is very important in the pro-inflammatory cascade by the generation of, in effect, a chemokine activity. So I think there's a lot of potential for those drug classes, and that paper I was just talking about was just recently published. Suggests that would be a rich area when people find the right molecule.
I was just wondering how the antioxidants work as far as these reactive species, and does that then affect any of the other kind of oxidation-dependent reactions like bactericidal, like neutrophils?
Well, it could, and that's a key question if you're interested in drug development. Or are these going to be general immune suppressions? Are you going to have animals or people, more importantly, getting a lot of infections or have compromised immune response? And at least thus far, that hasn't been a problem. That a lot of the oxidation pathways are stress responses, normal responses. There's a lot of compartmentalization of these responses. The cytosol generally and the extracellular space, as a matter of fact, is very reductive, and so you have to have some very significant compartmentalization to allow these things to go on anyhow. But I suspect that's part of how it's tightly regulated. What has been a pleasant surprise thus far is that these animals nor people have not shown general susceptibility to infections or cancers or other things. Yeah, but it's a critical problem that hasn't been seen in animals. We're just starting in longer term therapies with people, but that's an important question. May have to do with the selectivity, because as you could see that many of the important pro-inflammatory responses are not affected by these drugs at all. Anything mediated by E-selectin or ICAM as opposed to VCAM escape untouched. What will be interesting, for the reasons I showed earlier with the knockouts, the issue of giving any drug during pregnancy is fraught with hazards. But that certainly, if it gets to the point of licensing, I'd be amazed if it doesn't have that kind of warning. Hasn't been tested that I know of. That's a long answer, and maybe more you wanted to know. But thus far, so good.
Okay, well, thank you.
Okay, thank you. It's a pleasure seeing you.